All right. Hello. How you guys doing? Having a small technical difficulty up here. Will be fine. Just computer's not connecting to OneDrive like it should be. Um, but I'm going to go ahead and start, and we'll just download the PowerPoint from Moodle if we need to, and we'll get going that way. Just not the way I usually do it. Okay, so today I was prepping for our class and realized our syllabus had a little issue in it. And I was also going to run by you a proposed change. So I want to talk about that. Apparently, I decided to accidentally skip today on the syllabus. So I added that back in. So we didn't have a date for today. We now do. That's good. But I wanted to propose a change because I have to travel on the 25th. That's the Tuesday before Thanksgiving. And I figured there probably wouldn't be too many complaints for doing that class at a different time. So my proposal for you, and you can let me know if you have some feedback, would be to give you a remote session the Friday before Thanksgiving to go over the exam too. That puts it a little bit later. I could probably have them finished grading by next week, so I could probably provide that to you a little earlier than I'm scheduling this, but my impression is that not everybody feels like you need to be there for the exam to talk about it. I could pass it back to you, I guess probably the day before that on our normal class period on Thursday. And then, of course, I would do it live probably from my home office, and you could ask questions virtually if you wanted to. Could hop on a Zoom call if you wanted to do that. So I'd be open to doing that. How's that sound? You like that? Some thumbs up? Anybody have any hesitations? I mean, feel free to email me separately if you'd like to for that. But so for now, that's my plan. Theoretically, I could handle this class, but I'm going to have to cancel my second class to make the travel plans work. So on that Tuesday before Thanksgiving. So that's currently my plan. That'll keep us on track as usual, give you the chance to review that as usual, even if it's a little delayed. Okay, so hopefully that's going to work well for everybody. And so that'll be Friday. I will announce some sort of a time, and I'm probably going to be doing this for both of my courses. So if you tune in at the wrong time, it might be a little confusing. Like, wait, what? What are we talking about? Okay, but that hopefully I'll be able to make that entirely clear, and I'll give you some time frame to tune in if you'd like. All right, with that, well, speaking of, today we're going to be talking about wastewater microbiology. This is really the beginnings of secondary treatment, as we called it, whereas primary treatment we already talked about. It's kind of that physical removal of debris from whatever's coming into the wastewater. And it will also essentially remove, we'll do settling, the first round of settling, and that'll remove a bit of the waste in general. So let me check on my status. It looks like I'm finally connected properly. So here we go. All right, so secondary treatment. If you recall, we put a picture of the overall typical wastewater treatment plant where we had some primary sedimentation. This would also produce some sludge that we have to deal with, and of course some screening bar rack and stuff. And then we have what we call secondary treatment, which would be some sort of a bioreactor. You could do anaerobic, but eventually you kind of have to do aerobic at some point if you're going to discharge to a surface water. Then we have the secondary clarifier, and then perhaps some tertiary treatment or disinfection or whatever else. So maybe we have some other pieces, but ultimately the bulk of what we're doing is in this secondary process, which would include a bioreactor and something to separate those microbes back out. The reason, like we talked about, it's very convenient to have the ability to change something that is at the molecular level into something that is a particle. So the sugars, the fats, whatever we couldn't grab physically with any easy means, well, we let the bacteria eat those, and then they make cells out of them, or digest them otherwise, turn them into stuff that's no longer digestible and is fine to discharge, and then we can collect those microbes. So the secondary clarifier does that for us, and we're going to talk a fair bit about what's happening in this control boundary, in this control volume, we could call it. You see this highlighted section. So we want to know what's the math that can tell us about what happens in there, how do we handle it, how do we navigate it, how do we use these mass balance principles that we've been learning about to understand how much waste we're removing and how much bacteria we're growing. So each one of those pieces you can kind of tell from what we've learned before will be essential to understand the system and to model it properly. So we need to know something about how the bacteria are growing. That's where we're starting today. And by the end of today, we'll be starting to think about how this would look if we were to put mass balances to it, to compare what we call the substrate or the microbe food. This is really the waste stuff versus the microbes that are growing. So we're going to get into some of the definitions. What are these symbols that we're using, each piece here, as we get going. So some terms here. First of all, waste-activated sludge is a common way to describe exactly what's happening in this tank. We are activating the sludge with oxygen or air that has oxygen in it, and that's allowing it to eat a bunch of stuff, grow a bunch of microbes, and it's kind of results in a sludgy type mixture because it's a whole bunch of bacteria, particulate matter floating around. So it's kind of a slurry, microbes plus waste, whatever's in there, and we're constantly aerating it. That aeration gives us a situation where it's pretty much going to act like a CSTR. Sometimes we might have a very long channel, the whole thing is aerated, and maybe mathematically it makes more sense to model it as two CSTRs in series because you're not getting much mixing from the very front of that to the very back. But at least in segment-wise, we could treat it as a CSTR or maybe a couple. So typically we'll treat this as a CSTR. Another thing we'll talk about is the strength of the wastewater. That's really how much food there is for the microbes, is what we're saying as strong. You can consider how much biological oxygen demand, that thing we talked about last time, substances demanding oxygen, how much of that is in there. So typical wastewater would range on the weak side from about 50, 60 milligrams of biological oxygen demand or demanding substances per liter, all the way up to 600, 500 to 600 range would be very high strength wastewater, so-called. What this means is if we have 1 liter of water, and typically at equilibrium with the atmosphere, at this room temperature is a little chilly, maybe there's 9 milligrams per liter of oxygen. So 9 milligrams, and then suddenly you're demanding 60 from it, you're going to deprive the water of oxygen completely, and then you have to wait for some time to allow oxygen from the air to get back in. So there's an important balance there. We're going to study that a little further in terms of what happens when we discharge this to some lake or river. But the point is, for the secondary treatment, we're trying to get rid of that to the point where it's not going to demand it all from nature. Okay? So this is our BOD. We might have quite a lot of this demanding substances. Sometimes we'll have situations where there's not a whole lot. The South Baton Rouge wastewater treatment plant often has a lot of infiltration from rain, and so we often have a situation where it's actually rather weak. It's very diluted compared to some wastewater systems. Okay, other terms, we have volatile suspended solids, VSS. This is how we estimate how many microbes we have in the water. So this gives us a microbe concentration. We will use the term X here, and by the way, we'll term this, I have another slide detailing all of this, but we will term this S for substrate. And we will term the microbe concentration on a mass basis as X. Now, previously in the class, we've talked about microbes. After we've done that primary sedimentation and then we do this waste-activated sludge, we have so many microbes that whatever organic materials in the water floating around that is a solid, we'll just call that microbes. And that gives us a good estimation of how much microbes do we have in the system. So looking back for a moment, you'll see we have in our aeration tank a volume, a microbe concentration, and a substrate concentration. So these are the pieces that we're kind of building for how we bottle this. Okay, the way we do this is we take some water or some sludge or whatever it is. We filter it to grab the solids. Then we dry it, just get rid of all the water mass. We weigh that, and then we burn it and see how much volatilizes. After we burn it, we only have some ash remaining. We take the difference between the dry weight and this ash that's remaining, and that's all the stuff that volatilized and went away. So anything that could transform into CO2, gas, nitrogen gas. So if you think of like a sugar molecule or a DNA molecule or something, most of that will volatilize. There will be just a tiny bit of residual left. So any metals that happen to be caught there, any inorganic particles that were floating around, those will not get counted as our bacterial mass, but everything that could burn away, we're calling that microbes. So it's not technically accurate, but it's plenty good enough. It's a pretty good way to do it. So that's the process, and that gives us X, our volatile suspended solids. And we'll often talk about it as milligrams per liter of volatile suspended solids, mg VSS per liter. Okay, a little bit about our typical outcomes. We will often remove 95 to 98% of these biological, biological oxygen-demanding substances, biodegradable substances, however you want to call it. Biological oxygen demand is the standard BOD. So we'll remove 95 to 98% of those substances. So that means if we started with 100 milligrams BOD per liter, we're going to end up with 2 to 5 range, right? So that's super helpful. And that's really dealing with primarily the carbon component of what's in our wastewater. There's actually a few things we might want to worry about in terms of what all can be a problem out in nature. The food is the primary one, but there's a few elements of food. So if you think about even our own diets, right? We think about nutrition more than just how many carbohydrates there are, which is maybe would map to the carbon. We also think about proteins and vitamins and other things, right? So if we consider what microbes need to grow, there are 3 primary nutrients, one being carbon, the next being nitrogen, and the last big one is phosphorus, although compared to those two, it's not very big. Compared to some trace elements, it is very big. So we have what we could call a CNP ratio that tells us something about whether or not our waste is limited by one nutrient or the other. So typical biological life has a CNP ratio of somewhere around 100 to 10-ish to 1-ish. These numbers change a little bit based on what kind of biological system you're looking at. This type of analysis is often used for oceanography to see, okay, will there be an algae bloom if we add a little phosphorus to this system? What's the limiting nutrient? But it also kind of applies to wastewater because we might have a scenario where we have plenty of this BOD, but no nutrients. In fact, when I was briefly a permit writer, I had a chance to visit a tissue recycling treatment plant. They were like taking cardboard and other fibrous materials. They were grinding it down, putting it into water, extracting all this fiber from it, and then remaking the tissue paper you might have like in a public restroom. So they're making all sorts of tissue products by recycling, and they're left with a process stream that had a whole bunch of carbon. Almost all of the waste was carbon. So because they were using all this fibrous material, they had tons of carbon, but almost no nitrogen and phosphorus. So what they had to do was they literally took fertilizer, like store-bought fertilizer, and they would pour X number of gallons into their treatment process every day to provide the nitrogen and phosphorus, which normally we try to remove those from our wastewater. We don't want to have those discharge in our wastewater, but to allow the microbes to grow, they had to add these nutrients. So I say all of this to kind of talk a little more detailed about this BOD. It's generally a good track of how much carbon there would be in the system. And in most municipal treatment systems where we're getting domestic sewage, there's going to be a good balance, and it's usually we focus on carbon, and then later maybe we have to do something to try to remove more nitrogen and phosphorus. This looks like I just put oxygen here, so there we go. Okay, so that's a little bit extra info to understand it. I also have a little video here we can pull up, and this will kind of give us a show of a typical treatment process. Just kind of taking a look at what they might look like. So this should be our primary effluent, meaning it's gone through our first sedimentation. Actually, this might be even the influent here. Yeah, I think that's probably the influent, and then this first chamber here would be the primary clarifier on the right. So doing some settling there. It's going to go across there, and then it will enter the aeration basin here on the left. So you see it, the water's coming over the top there. That should be the way we operate a sedimentation, right? Only the top of the water comes across, and nice and gently, and then it's going to flow over and into our secondary treatment step here. And again, I think I already told you, but you definitely don't want to fall into this, right? Because, well, for obvious reasons, but plus the drowning hazard, because you're not going to float. So all the bubbles, you'll have no buoyancy. So if you look closely, you can probably find some ladders or escape routes out of there. Yeah, in case it wasn't obvious, don't fall into a wastewater treatment system. And it's also probably like 18 feet deep or something. So it's you see that nice sludgy water there. This will come into the last part, which would be the secondary clarifier. We get some foam. There's some floaty stuff that was formed. That's pretty typical. So we get some flotation of things and some sedimentation of the rest. And then it's going to continue on. And then once it leaves that secondary treatment system, we're going into what could be a disinfection contactor, or not exactly sure what else they might be doing here. But so that's probably a chlorination chamber. And then we'll have effluent that now, if you look closely, kind of just looks like river water. So it looks a good bit cleaner. I think there's a better shot in a moment where you can see yeah. So that could be a river or stream in terms of just visuals, right? So that's a nice, quick little walk through. And I don't even know what country this is in. I think it's somewhere else. But quick little walk through what a primary plus secondary treatment system might look like. And that would be a relatively small treatment plant, I think. That's not a huge one by any means. Okay. So how do we know how much growth we have for these microbes? We want to know how fast they're growing and how fast they grow in reference to how much food they have. Because the faster they grow, the more food they're taking. Therefore, the speed at which we get rid of this food, the waste, is related to how many microbes we have and how fast they are growing. So there's a lot of terms here. We've already talked about several of them. And what we want to aim at is some rate constant for microbial growth. and to understand microbial growth in reference to our substrate use. So how fast are we consuming the food? So we have what's called the Monod equation here. This is giving us essentially a rate constant. We're going to call this mu, but this will be a first order rate constant. So mu is effectively the type of K that we've talked about before. It'll be a rate constant. I'm going to show you where it comes from and show you all the terminology, and you'll have this on your formula sheet, which I need to update for you for the final one. But we don't have to know every last detail about how it's derived. We're not even going to derive it per se. So I'm just going to show you where the pieces are coming from. And we'll spend a few minutes familiarizing ourselves with what this is and what the pieces mean. So we have this rate constant is going to be equal to some maximum rate constant that we could find if the bacteria were given every favorable condition they could possibly want. As much food as they could ever want, and the right temperature, everything. So this is the maximum specific growth rate coefficient, mu m. So mu max is the max, okay? But sometimes, in fact often, we're not at the max. And so mu is usually going to be different. We could actually look at when does mu equal mu max? Well, that would be when all of this is equal to 1. When does that equal to 1? Well, when S is big enough so that it's pretty much S over S. So this Ks, whatever that is, it has to be small compared to the S, and then our mu is pretty much at mu max. That's a little side note, so let's just continue on thinking about what each piece is. So S is the amount of food. Bless you. So that's our substrate. That's going to be our milligrams VOD per liter. Been talking about that. Mu, that's the specific growth rate coefficient in per time units. This is what happens to be the scenario for the bacteria in this moment, or under some conditions. Okay? So that we often will have to solve for that. Perhaps we're given it and we need to figure out something else, maybe something about the substrate. Okay, we already mentioned the mu max, the very most favorable conditions. Under those growth conditions, we will find mu max. Now this Ks, we're going to have many Ks. I'm following the book's terminology, so I apologize. But this is an uppercase with S as a subscript. This is what we call the half velocity constant. It's essentially the amount of substrate when mu is half of its max. I have a slide showing you what that means visually, but don't worry too much about this. So I'm going to explain it. I'm going to show you what this looks like, but this is just kind of background information here. So we have, if we plot mu against how much substrate we have, we can expect to eventually find the max. So this would be mu max here. Now, we would find that at a very high substrate concentration. Even if we just look at the equation, when S is very, very large, that's when we find mu max. And practically speaking, if you give microbes an infinite amount of food, that's when they can grow the fastest, right? The more food, the better. So if we have very little food, we can expect to start near 0 because they can't grow at all, right? So would make sense to start at 0 and start going upwards from there. And so this equation, when we plot it, ends up looking something like this. Okay? And we'll asymptotically reach mu max as we add more food. So what does Ks mean then? Well, Ks is when we take, we find half of mu max. Let's call that maybe here. Well, what is the substrate concentration when we are at half of mu max? So we would follow this along, find our value, and say, okay, here is then our, if I could draw straight, that then is our Ks value. So that's how we find it. That means we could do it experimentally. If we tried some values, found some curve, we can get that value. So mostly what I want you to know is this is an amount of food, and it has something to do with this halfway point for the microbes are halfway happy kind of thing. So don't worry too much about it. It's a parameter. We're going to need it. We'll either be given it, maybe we solve for it. It's a parameter, okay? And you need to recognize that this is a parameter that has units of the amount of food. If you look at our equation here, we have the mu equals mu max, which should be a rate constant, something like per time units, times the amount of food divided by two values that have food as the units. Okay? So it's rate constant, which is per time, times food divided by food is the units, meaning we just have per time. It comes back to a rate constant. So all of this means that mu ends up with per time units, just like mu max. So it's a first order reaction rate constant that we find according to that. So that's what models our growth kinetics, and that can give us some idea of how microbes will grow. Now, when we model, if we do this in a lab and we test it, there's a very nice, any of you ever seen Jovi? It's like a video journal. So instead of a scientific paper publication, it's a scientific video publication. So you'll have access to these through LSU's library. Links here. Pretty neat. They do a little video presentation describing all this. So if you wanted to know more or hear it again from somebody else, follow that link and check it out. But the essence here is if you give bacteria, if I put bacteria in a bottle and said, hey, here's some food, new environment, why don't you grow? It'll take them a couple hours before they realize, there's plenty of food here, let me shift my metabolism and start eating. So immediately you might have a little bit of growth, but it'll take a little while. We call this the lag phase before they really take off and start multiplying. This is, by the way, why we try not to leave leftovers sitting out, let's say in a warm sunny day in a picnic, something that's like nice and liquidy with lots of bacteria food in it. We don't want to let it sit out too much past a potential lag phase, because if there was some bad bacteria there, well, a few hours later they're going to start taking off, and they might be producing toxins, they might be just pathogenic themselves, and you don't want to eat them. Maybe a tiny bit of them is fine, tiny bit of contamination didn't hurt anybody, one or two bacteria lived through the cooking process, or maybe didn't cook it, so uncooked stuff. So you don't really want to give them that exponential growth phase, because that's when they start multiplying like crazy. And they have a doubling time of something like 90 minutes. So give them the first couple hours, probably not a big deal. That's our lag phase. Then we have some acceleration, and now we're in the exponential phase, or exponential growth phase. That typically lasts something like, so if this is, and this will vary depending on the bacteria, but since I'm talking about the kind of stuff you might be worried about at a picnic, we're talking about something like E. coli or salmonella. So the lag phase, maybe 3 hours, something like that. And then you have a growth phase that might last until, 18 to 24 hours, at which time you start to have a decline, at least in the net growth rate. You probably do still have plenty of growth, but now you are also having some death, because microbes have reached a point where they are now competing with each other for the food. It's no longer a case where there's an infinite amount of food, and our ratio of microbes to food has shifted from tons and tons of food, grow fast, to now, oh, we're all competing for it, and we're all getting old and dying. There's becoming a balance. And eventually we get to the death phase where there's a little bit more dying than eating and growing. So that's your typical profile if you just set up a batch with some discrete amount of food and let it run. So that gives us some indication of how these things are working. And if we were to do an experiment like this, we would start with some initial amount of X, initial amount of bacteria, some initial amount of substrate, and some oxygen. Then we can watch, if we check this over time, we could measure the substrate over time, or perhaps measure the oxygen. We'll talk a lot about that in later lectures, monitoring the oxygen over time. And we could monitor the bacteria over time. So we could watch how the substrate changes, ds dt, should be a decay of the substrate concentration. We could watch how X changes. This should be a growth equation. So we're growing bacteria over time, at least initially. And we could track all that. So we could use the same tools that we have been to track what's happening in terms of mass balance. Okay, so let's take a step further then and take this rate constant that we looked at from the Monod equation. This gives us that specific growth rate coefficient, kind of just K for a reaction. And let's put it into a reaction system, right? Let's put it into an equation. So if we have dx dt, we can say, since it's a first order equation, we could say dx dt equals mu x. This is just like when we said dc dt equals kc to the 1 power, right? So we've seen this form lots before. Now we're talking about it in terms of x, the microbe concentration, all right? So this is just parallel to that. And as we see, there's no negative there. So this should be a growth equation, and it's going to be first order. Okay, so we could describe then the rate at which we have growth and say that's mu x. And if we substitute mu into there, because we have that equation for mu, we could say that this is dx dt, or r of g, equals x times the rate constant, which is mu max times s divided by ks plus s. Okay? At least we didn't try to leave the rate constant as k. But as we'll see, we have other k's to deal with. Okay, so then, and this is, by the way, this is just simply substituting this to here. So we also want to know how fast we're using the substrate, how fast we're removing it. That's kind of the whole point, right? So we can relate the substrate utilization rate to what we call a yield coefficient. How many bacteria do we gain per waste that we consume? And that's a really important thing. So we can say we have a yield coefficient y that's related to the substrate utilization rate. That would be our k, in the following manner. And this feels a little backwards, but we're going to define this substrate utilization rate constant, or rate coefficient, as the maximum possible consumption divided by the yield coefficient. Guys have questions? Okay. Oh, perfect. Uh-oh. Have you tried it, or a little scared, Tina? So yeah, so anytime you have yogurt or something like that, these same principles are applying. So and feel free, like, I know I don't love it when people are just chattering about anything, but I love it if you're chattering about the class. So just speak up louder. And if you open it, by the way, you'll you probably hear like the pressure hiss. So kombucha, all that, it's like you can tell that, you know, fermentation reactions. There's actually a big connection between the way we create beer or other alcohol products to environmental engineering. So I have a lot of friends from grad school and stuff that ended up doing their own microbrewing and stuff, because it's like, yeah, it's this stuff. It's like, well, how long is it in there? How much food does it need? And all that. So if you open it, you might hear a, because all the gases they're producing, the byproducts from what they're eating. So good luck. Yeah. In that particular case, maybe those microbes are out competing anything else that might be bad, but they probably got to the point where they've kind of consumed all the whatever would be keeping them normally happy. So I don't know what to say. I don't think I would eat it at this point. Yeah. Anybody have crazy food microbe stories? Random. Okay.
And bottled it and then left. And then my parents were at home and my mom started to hear like explosions.
Oh no.
In the closet and the bottles were
bursting.
Pops off of them.
Yeah.
Something he did wrong.
Yeah, if you don't get it right.
It didn't ferment it enough or something, so there's still active fermentation happening in the bottles. Like there wasn't, the alcohol wasn't enough to kill what was left or something or the other.
Yeah. Yeah, there are moments where you have to stop the fermentation altogether or something like that. Sometimes you add like a yeasticide, something that kind of kills or stops the yeast. Because, yeah, if they keep going and they keep producing CO2 and whatever else, boom. So watch out with that bottle. Mason, you might want to move up. Yeah, no. I'm kidding. Shaking it. Can you squeeze it? Like, does it feel solid, or is it like, yeah, it's still a little squishy? It's not at risk of exploding unless it's like bursting, right? Yeah, it's like, could start swelling. Okay. All right, good practical aside here. All right, so this substrate utilization coefficient is going to tell us how quickly at the maximum can we use the waste. So it's going to be related to the mu max divided by this yield coefficient, and this yield coefficient is simply a ratio of how much bacteria do we get mass per mass of food, and we also add per day. So if you give microbes a day of munching on yogurt, how much bacteria do you yield per gram of yogurt munched, right? That's kind of the situation there. So again, a lot of these are just parameters, and we'll just have to sort through them on terms of what do we need to put in our rates properly, and we could see how we could measure some of these in a lab or observe them in some ways to get what's happening. Now keep in mind, these are two different Ks here. This is a lowercase. This is not the uppercase Ks. You'll have both of these on an equation sheet, so it's well worth spending a little time to memorize, make sure you understand each of their units and that they're different, right? So that would be a good practice as a study practice once I post the formula sheet, or just come back to this page and recognize that these are different and know that difference, and that will help a lot. Okay, so this means we can get a rate at which substrate's being removed, being utilized, according to essentially relating it to how much we're growing. This will be an inverse, or rather a negative correlation. So the amount of substrate we use will be proportional to the negative amount of how much bacteria we're growing. So growth means consumption of the food. We'll also divide that by this y, which is probably the best definition here of y. It's that ratio, how much substrate do we get per food consumed. Okay, so we've got the K will be some milligrams BOD per milligrams VSS per day, and the yield coefficient is milligrams of microbes per milligrams of food. per liter. And both of those are in per liter, so the liter drops out. Okay, so then we could say if Rg is mu x, and now we have R of the substrate utilization is negative Rg divided by y, well, the last piece that we need is the rate at which they die. So we have the rate at which they grow, we have the rate at which correspondingly they're using the food. The last piece we need is the rate at which they're dying so that we can capture all the different pieces we were talking about here. So that means we have to define Rd, and now we have a third rate constant and a third K. So this is Kd for the death or decay. This will be another rate constant, first order. This will be a decay reaction. So we have two reactions the microbes are going through with, a growth rate and a decay rate. So this will be negative Kdx as the overall rate. That's pretty self-explanatory, and again, we will just have some observation that gives us a rate constant for Kd. We don't have a fancy formula, we just have a Kd. So that's usually going to be given, maybe sometime we'd solve for it. So the question then becomes, what is our net growth rate? Well, it's relatively simple to calculate a net rate because you can have the rate of one plus the rate of the other. And in this case, that would be a growth plus the negative decay. At some point, those might balance. That would be our stationary phase. When the death and the decay are balanced, we reached kind of an equilibrium between those two, whereas R of G is predominant here and Rd is dominant here. Okay, so pretty straightforward. It's negative Kdx. We've done a lot of first order decay reactions, so that should be pretty familiar. The net growth rate, we can say R prime, the prime for net, would just be simply Rg plus Rd. The negative is built into the Rd here, so that would end up giving us Rg, which is our substrate mu times x minus Kdx, right? This is what we could translate that to. We can also describe the growth in terms of substrate. So we could substitute and say R of G is also known as the negative rate at which substrate is utilized times y, and put that in here, and we get negative y times RSU plus a negative Kdx. So there's a few ways we can organize, reorganize to get this. And this piece, the nice thing about it is it's putting things in terms of x and all these different parameters that we have. Okay, so that can give us a nice summation way to look at it, to look at the rates here, so that we can work towards some mass balance. All right, so let's do that. This is going to be more complicated than you're ready for at the moment. Technically, you could do it, but we're not going to accomplish this I'm not expecting you to accomplish this just on the fly right now. But I do want you to start it and see how far you get and how you're thinking about the mass balance approach. I think that'll help you to wrap your mind around where we're heading with this. So we're going to take a look at this problem. And by the way, today we are looking at a problem where we are just looking at the growth chamber. Next time we'll look at the recycle, and we'll notice that the math becomes, that recycle becomes very important, and it actually ends up simplifying a few things for us. But right now I just want to show you how these growth equations and other equations are going into the mass balance in a way that, based on what you've learned so far, you can do. This is just going to be a bit complicated and a little counterintuitive, so I'm going to walk through that with you. But you will see how the mass balance stuff that you've learned applies directly. Okay? So this problem, we have a table with some common parameters given, and you'll see we have a parameter for K, the maximum specific growth rate coefficient, substrate utilization coefficient. Mu max, that's the maximum growth rate coefficient. Ks, the half velocity constant. The Y, that's the substrate, the yield coefficient. And Kd, the death coefficient. We have a default value for each of these. I think the problem assumes we will use those. But in case you're interested, in our book has this table, and those are typical values. So in this example, the shallow pond depicted here stays well mixed due to the wind and a steady flow of water from a small creek. And the problem essentially asks, how much bacteria do we have after this waste comes in? It's well mixed. It's treated like a CSTR, essentially. The question that I think I probably cut off here is, how much X do we have after the pond, and how much S? So what is the resulting microbe concentration and food concentration? And these would be the final, or what's leaving. Now, of course, in a CSTR, we know that since it's well mixed, X in here is equal to what's leaving, and S in here is equal to what's leaving. So the picture kind of gives us everything. So the picture plus that gives us all the pieces we would need in terms of the parameters to solve this. We're given a volume, we're given a flow rate. All of that is in here, just simplified. And one thing you'll notice is there's no bacteria coming in. X naught is 0, which is a reasonable assumption if you have kind of a stream that doesn't have a ton of biological matter in it. That's probably kind of typical. So, here's the actual question for us. So the question says, the microbes in the pond consume the following biodegradable organic matter according to the typical kinetics. So we'll use those default values. So A, and so then we have to determine A, the BOD leaving the pond, that's our SF. B, the biodegradable organic matter removal efficiency from the pond. That'll just be S naught minus SF divided by S naught times 100%. So once we find SF, then we can find that. Plug in the number. And then C, the concentration of volatile suspended solids leaving the pond. And this is XF leaving. Okay, and I'm going to go ahead and read you the beginnings of the solution, and then from there give you a few minutes to think about how you would take the next steps. I'll walk you through this, and we'll derive an equation that I'm going to go ahead and give you on the formula sheet that is specifically for the pond type of scenario where there's no recycle. So we're going to derive something that I'm going to give you as an equation that is only useful in a specific circumstance. So when you're studying your formula sheet, that's the piece you need to know, is when this is applicable. Because if you try to use it with a recycle system or with a system that has some bacteria concentration entering, it's the wrong mass balance. You've got the problem wrong when you try to use that equation, okay? So the pond can be modeled as a steady state CSTR because it's well mixed and we have this flow. We're going to assume it's steady flow and steady composition for a long period of time, so it reaches steady state. Good assumptions. Quite convenient. First, set up a mass balance on the microbial mass using the pond as the control volume and the variables that were provided. And when we do that, which is, by the way, this is the backwards part, or the first backwards feeling part, is to solve for the amount of substrate, we're doing a mass balance on the microbes. That feels weird. As you'll see, it works out nicely that we have some of the X concentration in terms of S, right? The amount of microbes we have depends on S. So here's the counterintuitive part. We're going to start with a mass balance on X. So to solve S, we're doing a mass balance on X. So give that some time. In fact, I'm gonna grab the. I'm just gonna edit my PowerPoint because I've been meaning to do this. I'm gonna copy these default values here and place them there for you to work on. So go ahead and start working on that. I'll have a quick update for you in a moment with that. Yeah, feel free to work together, guys. All right. All right, there you go. right, I'll give you about 5 minutes to work on this. Like I said, please feel free to work together. Ask me questions along the way if you like. Just brainstorming time. All right, so I hear some productive murmurings over here. Seems like some of you made some good progress, making some good progress, and probably some of you ready to tune out. Okay, so I'm gonna walk through this with you here. The mass balance we were told to start with, which is a little counterintuitive, trying to solve for S, so we start with X. But you'll see for this particular case where we have no microbes coming in, X naught is 0, and no recycle, that keeps X naught as 0, because when we recycle, we add X to the influent. In that condition, we start off with our mass balance, 0 equals Qx naught minus Qx plus V times the net growth rate. Okay, so in this growth, we have two rates that are affecting the growth. That's the growth and the decay. So just as a little aside, I kind of summarize what we talked about with that. And we can call that the growth x times mu, where mu is mu max, times s divided by ks plus s. The death is negative kdx. So we can extract x from both of those terms. And in this volume here, we have Vx times those two rate constants, right? So the mu max s divided by ks plus s minus the kd from the death rate. So this equation, what we see is it becomes quite convenient when we rearrange to say q times x, we add that to both sides, equals Vx times all this stuff. We notice that there's an x on both sides that's simply going to cancel. That gives us a situation where we can cancel those, and now we have an equation that has s as our only unknown, and we can solve for s. And so from here, it's a little bit annoying algebra-wise, but we can certainly do it, to get an equation for s as a function of these other parameters, now that we've gotten rid of the only other piece that is changing, which is x. So it's quite convenient, a bit counterintuitive, which is kind of why I primed you the way I did. But we can march forward here. So the other thing we can do is we could go ahead and divide both sides by q. That'll just put it into theta instead of v. So this will be 1 equals theta times mu max s divided by ks plus s minus kd. So that's relatively straightforward and simple. And if only we were solving for theta, that would be quite convenient. But we need to expand this out and solve for s. So I'm going to step through that, and we'll find a final equation for s equals some function of these parameters. Okay, so this is just algebra from here, and I wouldn't even printed out my old copy of this to make sure I could easily correct a mistake if and when I make it. So we're going to factor that theta across. So 1 equals theta mu max s over ks plus s minus theta kd, bless you. And we're really just looking to solve for the s's, so we're going to put the theta kd on the other side. 1 plus theta kd equals theta times mu max s over ks plus s. From here, we'll go ahead and multiply both sides by the ks plus s. Like so. Get that equation, and we can multiply across so that we can get all the s terms by themselves. So we'll have ks times 1 plus ks times theta kd just doing the opposite of factoring out here, plus s times 1 plus s times theta kd. All that equals theta mu max s. We'll continue this with a little space over here. So we'll continue here and say, essentially subtract the S terms to the other side. So we will be left with Ks plus Ks theta Kd on the left side, and on the right side we'll have theta mu max S minus S minus S theta Kd. And now we can factor out the S's on the right side and get a term for just S. Okay, so then just rewrite this part. That'll be equal to S times theta mu max minus 1 minus theta Kd. So at the end of the day, S will be equal to Ks times 1 plus theta Kd. If I factor that a little bit, divided by theta mu max minus 1 minus theta Kd. Okay, and there is our equation for the substrate concentration. Let me do that a little better. And this is specifically for the no recycle case with 0 microbes in the influent. This is what we derived for specifically this mass balance, a pond type of situation where we have a CSTR with no microbes coming in, no recycle happening, therefore no microbes coming in, and this will be generic for these different thetas, Ks, Kd, mu max. What's key here is this is no recycle. We will deal with both. Right, next time we're doing more recycle, this time we're focusing here. So that gives us a way to solve for part A and therefore for part B. Question kind of then becomes, what is the approach to part C? Before I get there, does this make sense? Some sense? Yeah, question?
So when it comes to like, well, first off, so you're doing positive for the reaction equation because it's at the end of the day it's a growth, right?
Whenever we are adding a reaction term to our mass balance, we always add the reaction, and if it happens to be a decay reaction, that'll be caught up within the R equals, it should be a negative for a decay. So in this case, the net growth rate could end up as a net negative, but we will have the growth minus the decay within that. So that's our, when we write it here, we say, and that's a good question, we say it's the growth rate plus the decay rate, and then the negatives caught up within that decay rate.
And then, sorry, right. So when it comes to actually solving for S, I always seem to mix up whether or not I'm putting like S naught in that reaction equation, or if I'm putting S final. You know, I don't ever always mix it up. I don't know which one to do.
Okay, so in this particular case, we did a mass balance on X. So our starting point had no S naught. This is a weird case. So this, if you were struggling with C versus C naught previously, this is a more complicated case. So just bear that in mind. But when we started the equation here, we set up a mass balance on X, so we have some amount coming in. That's where we get the initial. So that's where our X naught, if we had any, is coming in this term, which is the amount of water coming in, bringing in X. Later, when we're dealing with X that might be leaving or might be what's being reacted in the reactor, that's going to be X final in a CSTR.
You wouldn't be able to set up the mass balance just in terms of S naught and S final, I guess, just because the monokinetics are all in terms of concentration of X.
Well, it's a good question. Could we just set this up as a mass balance on S in the first place? let's just check what happens, because that's the start of the next problem, is to do that. But let's see for ourselves what happens. Can we isolate this from x, or do we need to solve for x first? Or rather, do we need to solve for x or s first is kind of the question that's not so intuitive, right? So we did we started with the mass balance on x to solve for s. Now let's do a mass balance on s. We're not going to finish this today, but we can start by saying at steady state, it's kind of our assumption, we're going to have some amount of S naught coming in minus some leaving, right? So we have those two parts. Then we're going to have some rate at which the substrate is being utilized, RSU. So we can then go back, flip backwards to our notes about how we defined RSU. We said that was going to be the negative of our growth rate divided by y. I'm going to flip back to double check myself there. I'm pretty sure that's the arrangement. And we talked about that right here, because we can define the rate of growth in terms of how much is being used. We rearrange that just a little bit, and we get this equation. So we can say then, and this will be in that volume. So as we proceed here, we need something to do with the RG. And we said that was mu times x, a growth equation, right? So this is going to be, R of G will be the mu max S divided by Ks plus S. Okay, so then for the R of substrate utilization rate, this will be negative that divided by y. So I'm just going to go ahead and pull this and say x over y times mu, which is mu max S divided by Ks plus S. Okay, so we can take that rate of substrate utilization rate, put that in here, and now our mass balance, what we're looking at, has S, and it has X in it, and when we start piecing it together, there's no obvious way for something to cancel in this scenario. So X is a variable, S is a variable, and unless we solved for one of them already, we have two variables in one equation, right? Two unknowns, one equation. So that becomes quite difficult. So let me just write out the next step and then answer any last questions. So we'd say 0 equals Qs naught minus Qs plus V, and I'm just going to fill this in here, times negative x over y times mu max S over Ks plus S. So what you see is if we solved for S already, we can get this equation into a just solving for X. Pretty straightforward. And that's kind of the task there. Any other questions? Does that answer your questions? Good? Okay, so we'll probably finish that out next time, and we'll definitely talk about the recycle system, which uses different mass balance next time.